Hi there. During aging, senescence generates many types of pathology, some of which contribute to organismal death. But what is organismal death, and how is it triggered by senescent pathologies? The process of cell death has been intensively studied, but organismal death hardly at all. We have been studying this in the nematode worm C. elegans. We showed before that when C. elegans die, something remarkable happens. If you look at them under ultraviolet light, a wave of blue fluorescence passes through their intestine, usually from the head to the tail. This death fluorescence is caused by a wave of necrotic cell death that flows down the intestine. Cellular necrosis is a self-destruct process where increased calcium causes lysosomes to burst, releasing protease enzymes that destroy the cell. Here's what the death fluorescence looks like. But what triggers this wave of death in elderly worms? We have now discovered that when worms die, there is another head-to-tail wave of muscle hypercontraction. This wave, which we call death contraction, is closely coupled with the wave of death fluorescence, but happens just before it, as you can see in this time-lapse movie, which is speeded up about 100 times. Here the muscle appear white. You see how the wave of muscle contraction moves along the worm. And now starts the death fluorescence. And now the worm gets longer again. This figure shows a change of relative worm length during death in young adult worms killed with oxidative stress. It decreases and then increases again. And here you can see that death contraction passes down the worm from anterior to posterior. Such death contraction followed by recovery resembles the process of rigor mortis, which happens in mammals shortly after death. We then showed that death contraction is caused by muscle contraction. It doesn't happen in worms that can't contract their muscle. In mammals, rigor mortis is triggered by ATP depletion, which leads to calcium release within muscle, and this causes muscle hypercontraction followed by proteolytic destruction, which makes the muscle go soft again. So rigor mortis is caused by muscle necrosis. By using in vivo sensors of ATP and calcium in muscle, we show that death contraction coincides with a big drop in ATP and a wave of calcium release in the muscle. Interestingly, long-lived DAF2 worms are resistant to death contraction. This raises the possibility that resistance to organismal death contributes to their longevity. Here is a cartoon showing how it all happens. Death contraction starts in muscle in the head and then moves along the worm, destroying the muscle and triggering a wave of intestinal necrosis. So, dying C. elegans get rigor mortis. But what's really interesting is that instead of getting it after death, they get it near the beginning of death, perhaps because unlike mammals, they don't die from cardiovascular failure. They're so small, they don't need a cardiovascular system to get oxygen. This work reveals some of the biological mechanisms at the border between life and death that occur in animals ranging from worms to humans. Mechanisms of organismal death are one link in the chain of events leading from healthy adulthood to senescent death. A critical question now is, how do senescent pathologies lead to organismal death?